Good afternoon. Welcome to RSSL's second webinar in our sterile manufacturing series. This one is reviewing sterile products and examining the factors required for release. Before we start, just some little housekeeping tips. If you've got any technical issues, please put a short message in the questions box on the right-hand side of your screen. Our team will then respond to you direct and try to resolve your issue. Please be aware that we're working remotely, so there might be some delay in solving your issue for you. During the webinar, RSSL would like to invite you to ask as many questions as you want, also using the questions box on the right-hand side. At the end of the webinar, time permitting, we will try to answer as many of these as possible. Any we don't get round to, we will answer offline and share them with you all, along with the slides from today and a detailed white paper produced to supplement this webinar. For those of you who attended the first webinar in the series back in April, welcome back. If you didn't attend but are interested to learn more, it's still available on our website to download on demand. If you are tuning in for the first time, my name's Annette Russell and I'm the Sterile Manufacturing Lead at RSSL. My role is to help facilitate our clients' requests, supported by a team of experts in the field. If after the webinar you would like to learn more about our offering, please contact me direct via email or the telephone number that you can see on your screen now. I'm sure, like us as RSSL, you've all had to learn new ways of working and living due to COVID-19. We've all had to adapt and learn to live life with social distancing, both around our business lives and our home lives. Here at RSSL, our support staff, such as myself, are now working remotely, which has its challenges, I'm sure you know. To break up my workday from my home day, I've returned to my crocheting and chose to replicate my own virus. Next, I'm going to work on crocheting my own testing kit and vaccine to go with it. On a serious note, our analytical staff have been working very hard during COVID-19 to continue to provide the services our clients expect. RSSL have been involved in helping out those clients whose drugs are needed in the frontline support for patients, as well as those who have had to escalate their research to get a COVID-19 vaccine into clinical trials. The whole of RSSL have been involved, especially our sterility and microbiology team. If you attended the first webinar, you'll know that we were very excited to have expanded our service offerings in 2020 to include a newly certified sterility suite, which is at the front line of testing those drugs for patients with COVID-19. Those of you who met the team last time will know that they're a fun team with a serious objective to support our clients, both non and sterile manufacturers, to the highest possible standard. We do this by offering endotoxin testing using the LAL method. From last quarter, we were able to offer sterility. We have mycoplasma coming this year with fast mycoplasma testing by the end of the year. And we can support all your raw material testing, as well as your routine vial and stopper testing. And our team of experts can help with those more complex projects, such as investigative foreign body projects. We can help isolate, identify them, and can even set up a reference database for your fill finish lines that we can refer back to. We can also help with any cleaning validations you may be interested in and environmental monitoring. And RSSL can offer support by our training and consultancy group who can help advise on those topics and more. I would now like to introduce you to our guest speaker, Dr. Tim Sandel. I'm sure many of you have heard of Tim and subscribed to his many publications. Tim has over 25 years of experience of microbiological research and biopharmaceutical processing. Tim is a member of several editorial boards and he has written over 600 book chapters, peer-reviewed papers and technical articles relating to microbiology. Tim works for a pharmaceutical manufacturer in the UK and is a visiting tutor at both the University of Manchester and UCL. I'd like to hand you over to Tim now, who will take on the rest of the webinar.
Okay, so hopefully everyone can see my screen okay. And we shall begin. Okay, so this is what I'm covering today in this session. So we're going to have a look at some of the key considerations for the batch release of sterile products. This is both aseptically filled and terminally sterilized. So we're going to be considering why sterile products are at risk, some of the key parameters for sterile products, the importance of sterility and sterility assurance, the important points to consider for batch release. And then we're going to look specifically at some of the key issues for terminally sterilized products and some of the important focal points for aseptically produced products. And as we run through this, the important message is that it's not just what is written in the batch record that is important for batch release, it's a wider view of the whole sterility assurance process. Now, sterile products are a particular risk. So not only do we have concerns about how well the medicine works, we also have the additional concern that if we don't get sterility assurance right, we could well be harming the patient. So this is why there's this additional focus on sterility assurance for batch release in tandem with issues around drug efficacy. Now, the presence of microorganisms in pharmaceutical preparations can reduce the efficaciousness of the product or make them unsafe for the patient. And in relation to the severity from the risk of contamination, and due to the route of administration through which sterile products are delivered to the patient, such as intramuscular or intravenous or certain inhalation products, this is why sterile products are at greatest risk from microbial contamination and why those risks are more impactful on the patient. So there are two broad methods to produce a sterile drug product. The first is terminal sterilization. And the terminal sterilization process involves filling and sealing product containers under high quality environmental conditions. And these are designed to minimize microbial and particulate contamination of the product. And here we have technologies such as moist heat sterilization, irradiation, and for medical devices, ethylene oxide. Aseptic processing of sterilized components. The drug product containers and closures are all sterilized separately, and these are then brought together under grade A environmental conditions. Now, there are several reasons why the focus on sterile products release is important. And the number one is always going to be the safeguarding of patient safety. So that's always paramount. But pharmaceutical manufacturers always wish to avoid recall. They wish to avoid suspending manufacturing. There's also the issue of reputational damage. There's regulatory concerns because a sterile product's recall would most likely lead to a inspection. And there's also the economic cost of not producing for a period of time. Now, the key prerequisites of sterile products are that they are sterile, and that's defined as the absence of viable microorganisms, which is something that needs to be assessed in terms of probability based on overall control of sterility assurance. There's also a requirement that sterile products are pyrogen-free, or at least containing such a low level that they are apyrogenic, and they also need to be free of visible particles. Other key quality attributes of sterile products are their chemical and biological purity, that they have the correct dose and strength and they are correctly labelled, that they appear as they should in terms of colour, particle size, viscosity, and so on, and there are no physical contaminants. So these are all some of the factors that need to be accounted for. It's also important to get an understanding of what we mean by sterility. So I said earlier, we're talking about the absence of all viable microorganisms. So something can only be deemed sterile only where there is a complete absence of microorganisms from it. And importantly, sterility is an absolute term. Either something is sterile or it is not. There is no such thing as slightly sterile or almost sterile. Another important concept is that sterility cannot be tested. We can only validate processes, build around controls, and adopt a science-based approach to risk assessment to help build in sterility assurance. And sterility assurance should be seen as a series of layers that need to be unpicked. And the sterility assurance system is a term to describe the sum total of all these arrangements that will help assure us of the sterility of our products. Now, with the release process of sterile products, then the finished product and the batch and all the associated data require review, and this initially undertaken by the quality assurance function. And batch review is not just about the review of compliance to good documentation practices, and it's beyond just checking the critical process parameters and the critical quality attributes assigned to the product being manufactured. We also need to understand the totality of elements that go into supporting sterile manufacturing. Now, arguably, batch records can be improved by the transition from manual records to electronic records, provided such electronic records have been appropriately qualified and they meet the data integrity requirements that relate to computerized systems. Because to a degree, the use of electronic records allows more data to be captured and assessed in real time. Now, the person who is responsible for certifying that the batch record meets the necessary requirements needs to be responsible for ensuring that all relevant aspects have been met prior to issuing the certification. And this individual will have a responsibility to ensure that no product is released prior to the legal requirements of GMP being met. And this person who's undertaking this task must have good knowledge that the relevant quality systems are in place and be satisfied that the product is fit for use, that it complies in terms of its marketing authorization and will not put patients at risk due to inadequacies around safety, quality, efficacy, or quality control. And to do that job in relation to sterile products, then the person needs to have a thorough understanding of sterility assurance issues. Now, there is a key difference between the US and Europe. In the US, the batch release is normally instigated by the person responsible for quality control, whereas in Europe, there is a system for qualified persons. So with batch records, now batches are generally produced with reference to a master formula, and this can take the form of a document or a series of documents that specify the starting materials with their quantities and packaging materials outlined, together with a detailed description of procedures and the precautions required to produce a specified quantity of a finished product, as well as the processing instructions, which also extends to in-process controls. And the batch is assigned a number or a lot number. And there are some general points for any batch record, be that sterile or non-sterile products, that are important to have in place. So we need to have the identification of the product name. We need to understand the weight, measure, or count of each component used in the manufacture of the batch. We need to know what equipment was used and whether that equipment is qualified, all of the components. We need to have an understanding of the yields at each manufacturing step and the expected yield of the finished product. We also need specific instructions for the state of the manufacturing process, the sampling and testing procedures, and any instructions that are going to give insight into any manual procedures. And with the concept of a batch, this refers to a specific quantity of a drug or other material that is intended to have a uniform character and quality, and that's produced within specified limits and produced according to a single manufacturing order and operating the same cycle of manufacture. And importantly, a batch refers to the quantity of material, but it does not directly specify the mode of manufacture. And with the production of a batch, GMP requires that a detailed record be kept of every stage of the manufacturing process, also detailing all of the required testing and also looking at the release process. And sometimes different terminology is applied to these documents, but in all intents and purposes, batch record is the element. Now, there are some other important aspects as well before we move on to sterile products specifically, to take up the remaining main part of the webinar. There are some general things that are really important to observe to avoid regulatory issues or the dreaded recall. And it's important to check that all entries are complete, including signatures and dates, to ensure that all necessary batch processing limits have been met, and that any deviations identified where limits have not been met have been called out and closed off. And it's important here with deviations to look wider and consider any other batches that might be implicated from a particular deviation. It's important to check that calculations have been performed in line with agreed formulas and the calculations have been correctly performed. We want to know that the operators have been signed off to show that every step has been completed by people who are appropriately trained, and where we require independent verification, that has also been completed. We want to ensure that any corrections have been made in line with good documentation practices. We want to check that all specifications have been checked against results, that printing materials are legible, and they, quite importantly, detail the correct dose strength, that any controlled changes have been fully assessed and risk assessed in line with the batch. Any additional sampling required has actually been carried out. Importantly, that finished product test results are in compliance and all quality records in association with the batch have been closed off. But of course, with any system, errors can occur, and that stands equally with batch records as well. And although errors can occur, it's very important that such errors are captured during the review process. And some of the common issues that can impact on batch records include incorrect data entries, failure to recognise out of specification results, missing signatures, writing out the incorrect form, or leaving forms incomplete where key information is required, or sending forms off to the wrong people and batch records actually going missing. So there are multiple reasons why batch records might not be completed properly, or they contain errors, and a lot of that comes down to their design. And it's important that all batch records are designed with the operators in mind. And again, electronic records can assist with this because it makes it more difficult to move on from one stage to another stage unless a key entry has been completed. Okay, so now we're going to turn our attention more specifically to sterile products. And we begin by having a look at terminally sterilized products. So for terminally sterilized products, the key aspects are the product design, having good knowledge and control of the microbiological conditions of starting materials and anything that goes into the process, such as gases and lubricants. We want to control the contamination of the process of manufacture, so we're avoiding the ingress of microorganisms and factors that might promote the multiplication of those organisms in the product. And this can be supported by cleaning and sanitization of product contact surfaces, the prevention of aerial contamination by handling under appropriately controlled conditions or use of barrier technology, the use of appropriate control time limits, and controlling time is a key thing about reducing the opportunities for microbial proliferation, and where necessary, filtration stages. We want to prevent any mix-up between sterile and non-sterile product streams. We want to try and maintain product integrity and have a thorough understanding of the sterilisation process. And to inform that, then risk management is of great importance. And where possible, as much data as possible should be obtained through real-time monitoring to enable the batch to flow between stages and have confidence that as the batch moves between stages that we have good control. Now, if regulatory approval is given, terminally sterilised products can be released through parametric release. And this is a system of release based on information collected during the manufacturing process, which is verified and compared against validated parameters. So parametric release is based on evidence of successful validation of the manufacturing process and appropriate review of documentation. So in essence, parametric release has come to mean the release of sterile products without recourse to the pharmacopoeial sterility test. And importantly, dropping of the sterility test can only be applied to terminally sterilised products. It cannot be applied to aseptically produced products, and with terminally sterilised products, only to those products where regulatory approval has been given, and regulatory agencies vary globally as to which sterilisation methods they accept and don't accept. Now, with terminally sterilised products, the most important parameter to consider is the pre-sterilisation bioburden result. And this bioburden assessment should be performed for each batch. And the sampling locations of filled units prior to sterilisation should always be based on worst case scenarios as drawn from validation studies and be representative of the batch. So for example, we're running a moist heat sterilisation process, for example, we want to be aware of where the colder spots are, and these are the samples to gain, or those locations of the bioburden levels. It's also important that we're not just going to become concerned with the count, for any organisms found during bioburden testing need to be identified, and their impact upon the effectiveness of the sterilisation process needs to be determined. And where possible as well, we also need to, or where appropriate rather, we need to have an understanding of pyrogen or endotoxin levels that also might affect the product. Now, with the release of terminally sterilised product, and I've mentioned validation earlier, these products must have gone through a validated cycle with appropriate thermal or radiation dosimetry with biological qualifications, and they should be capable of demonstrating at least a sterility assurance level of 10 to the 6, and these days often that's extended out to 10 to the 12. And this is reference to using biological indicators with a defined resistance to the sterilisation process. It's also important that we have an understanding of the integrity of the containment system, so we don't want to have the post-sterilised product subject to ingress of microorganisms. And that means qualifying through microbial challenges, and of course the assessment of the pre-sterilisation bioburden, as we just discussed, looking at counts and organisms, and spore forms are going to be our chief concern. Okay, we're now going to move on to aseptic processing. And aside from direct product tests, this is where it's even more important to have confidence around the background controls to aseptic product manufacturing. So performing aseptic filling, there are a number of variables that need to be defined and kept under control. And these variables involve different elements in the process, such as personnel gowning, their personnel aseptic behaviour, particularly how personnel perform interventions in the proximity or within an area where the product may be exposed to the environment, the operation of heating, ventilation and air conditioning and air filtration systems, such as through HEPA filters, maintaining pressure differentials between adjacent areas, the robustness of cleaning and disinfection procedures and practices, and how material gets in and out of different areas and different clean rooms. In this scenario, the monitoring of environmental contamination through viable and non-viable methods is also a critical activity. And regulators have progressively been putting in place stricter contamination limits within areas where aseptic filling takes place. So understanding these variables of control is essential for the person tasked with batch release. They've also got to know that the clean rooms were functioning properly and all background environmental monitoring was indeed suitable. Okay, we'll now have a look at some of the kind of tests that need to be undertaken and the supporting information that will inform the batch record. So to begin with incoming raw materials, the quality of the raw materials and active ingredients and excipients must have been assessed as coming from an approved supplier who is providing those materials according to the appropriate pharmacopoeial grade. Now, typically, quality control testing is performed and reviewed prior to release. And in relation to our topic of sterility assurance, this will include the microbial limits test. Other information that should be presented includes records tracing the use of the materials, so where have they been used in the manufacturing facility and for which batches, documentation of the examination and review of materials for conformity, checking against established specifications, and a clear statement as to what the final decision has been regarding the release or rejection of raw materials. This also may apply to intermediates where we have multiple manufacturing sites, and also packaging materials as well. It's also important that we have an overview of in-process controls. So an important feature of the pharmaceutical manufacture of sterile products is the microbiological monitoring that is performed of the product at different stages as that product is being manufactured. So although such monitoring may not necessarily relate directly to the probability of sterility or non-sterility, such monitoring acts as a good indicator of whether something is going wrong, whether there's a drift or something that could go wrong. So trend data can also be quite important, and such data becomes more important the closer we move towards final formulation of the product and prior to final filtration. An important aspect of sterile products manufacturing is process hold times. And here it's important that time limits are established and justified for each phase of the processing period. And this extends from the start of the process through to bulk product compounding and through to final sterilization. And that's because the longer the time, then the greater opportunities there are for microbial proliferation. And it might be that cleaning validation, as applies to a vessel, might be good for 24 to 48 hours, or the pressure hold of a vessel may have only been qualified for a certain period of time. And such issues become even more important where our product is capable of supporting microbial growth, and that is it has the necessary growth factors and so on. So control of time and then associated things like temperature as well. And our main method for assessing at this point is bioburden monitoring, and most commonly this is around conventional total viable count techniques, which can be in the form of membrane filtration or as pour plate counts. We also need to understand how we've controlled our sterilization processes. So a number of elements that go into the manufacture of an aseptically filled product are sterile, either purchased sterile or sterilized in-house, and this includes vessels, holding tanks, manifolds, and so forth. So it still remains that even in the expanding era of single-use disposable technologies, that steam sterilization is the most common form of sterilization conducted within sterile manufacturing facilities, as would be conducted using an autoclave. And here the most important aspect in relation to steam sterilization is ensuring that all of the trapped air is removed from the autoclave before activation. And this is because any remaining hot air would act as a poor medium for achieving sterility at the surface level of the item we're trying to sterilize. And it generally follows that failure to sterilize, particularly in a porous load autoclaves, is almost due to air being trapped in the load. Another area that requires assessment is ensuring that the load is dry. For wet loads, not only signal problems with steam quality, it also stands that any wet load is a sign of non-sterility. So autoclaves should be periodically assessed using biological indicators together with thermometric data, and the industry norm is either every 6 months or once a year. Now, I did mention briefly just before was that of single-use disposable sterile technology. So some of the challenges around sterilization can be overcome with such devices. However, you do not only need an overview of the sterilization method and how robust that is, you also need to understand any reactivity of such devices in terms of leachables and extractables. An important element with aseptic processing is sterile filtration. Now, filtration in general will occur at different stages of the manufacturing process, but the final sterilizing filtration step is the key step required, which is the final point of transfer of the finished bulk directly to the filling line. And under EU GMP, this should be as close to the point of filling as practically possible. So unlike our earlier talk about steam sterilization, where we're trying to destroy or inactivate microorganisms, filtration is a means of sterilizing, in this case, fluids, but also would apply to a gas, by removing microorganisms. An important aspect of filtration is the pore size and integrity testing. Now, the porosity of quoted for filters is not obtained from direct physical measurements, as would be, say, to measuring the pores. Instead, it's done on the basis of the pressure which is required to displace liquid from pores. And this is referred to as the bubble point value. And this is coupled with a formula which takes into account the pressure required, the surface tension of the liquid, and the contact angle between the liquid and the surface of the pore. So knowing that our filter was integral before and after use is of great importance. And some of the main reasons for filter failure, and in this case filter failure often leads to batch rejection, are incorrect assembly of the housing, which occurs more frequently than it ought to, flawed or defective filter cartridges, a membrane filter, or the growth through of microorganisms under cases of particularly high bioburden. We also need to be satisfied that we have good deparogenation controls. So glass vials, ampules, used for aseptic processing need to pass through a dry heat tunnel as a key part of the process. And the regulatory standard for the qualification of deparogenation studies is endotoxin activation. And any device should be capable of inactivating endotoxin to at least a 3 logarithmic reduction based on a suitably high challenge of endotoxin. So that tends to be a minimum of 1,000 EU per device challenge. And this again is typically assessed at 6 monthly intervals. So in terms of the batch release process, it's important to understand that the temperature matched the validation and that the time that the glassware spends subjected to the highest temperature also is in keeping with the validation. Now, these days, most aseptic filling is conducted inside of an isolator, and this is designed to exclude personnel from grade A. And devices like isolators need to be biodecontaminated, and the most common, but not only form, is hydrogen peroxide vapor. Now, this is not classed by European regulators as a sterilisation process, and that's due to hydrogen peroxide vapor not having any penetrative ability, and also due to the concerns around occluded surfaces and sometimes fragility of the cycle. So there's an additional requirement that product contact sites, such as filling needle manifolds and stopper bowls, are separate to sterilisation processes. So key variables are the sterilisation of these devices and their aseptic transfer into the isolator prior to running of the decontamination cycle. Now, with environmental monitoring, there still remains little regulatory guidance as to the environmental monitoring program. But the people tasked with batch release must be satisfied that environmental monitoring has been conducted to a plan, and that the samples taken are in locations that allow an assessment of any risk to the product to be made, and that monitoring has been conducted at a sufficient frequency to capture every key activity. So in direct relationship to aseptic filling, that's every manipulation and every intervention. It also stands that the environmental monitoring techniques deployed should be comprehensive. And this is all about capturing sufficient data for the enumeration of environmental organisms using culture-based methods, or alternatively, rapid microbiological methods can be used, provided they have been evaluated to show equivalence or better with the conventional methods. It's also important with environmental monitoring that all excursions are investigated, and in relation to each excursion that the root causes have been determined. And this is particularly so for grade A excursions. And this will also include a corrective action plan when action levels are exceeded. And with grade A contamination, it's also important as part of the batch release process that a detailed assessment is undertaken, which also informs about the batch disposition and an indicator of whether the batch is suitable for release. It's also important to take into account monitoring outside in the area surrounding the isolator, be that grade B or grade C. And this is because that data, particularly if there's an upward trend, can indicate the potential build-up of contamination, say during loading the isolator or other activities. And this is where, as we mentioned in the first webinar in this series, the importance of taking a holistic approach to contamination control. The batch release process also needs to take into account water controls. And again, this is not data that will necessarily feed directly into the batch record, but we need to understand that our water systems in terms of generation and distribution are in control. And this means suitably controlled methods of preparation, storage and distribution, so that the bioburden and endotoxin levels are within specification at the point of use. And while non-batch specific water results are not always supplied with batch records, the batch review process needs to understand if there have been any excursions directly linked to the time of manufacture, and that these should be noted, particularly where there's a risk of endotoxin contamination from the batch. And we have the standard levels on the screen. That endotoxin WFI needs to be less than 0.25 EU per mil, and bioburden needs to be less than 10 CFU per 100 mil. And in practice, alert and action levels are typically set below these levels. But there are other controls to consider as well, and these can inform whether or not there has been a control breakdown. This might include the temperature at which the water circulates at. So often WFI is held at a temperature above 65 degrees, which allows it to operate in a self-sanitizing manner. If that's slipped, then again, that's information that needs to be fed into the batch release process and which may impact on the sterility assurance. The sterility test remains a key aspect of GMP microbiology, particularly for the release of aseptically filled products to ensure they are safe for use. But there are limitations with the sterility test in terms of the number of items tested and with culturability and what will grow or not grow within the defined culture media under the defined incubation conditions and those organisms that do not readily culture anyone. So this puts greater emphasis upon sterility assurance, and this is also doubly important for those products that need to be released early ahead of the sterility test result being available, as with certain chemotherapy products, for example. So here it's doubly important that the site's sterility assurance process alerts to the risks. The batch release process also needs to take into account laboratory data and laboratory controls. And laboratory records should be complete and the data derived from all tests conducted to ensure compliance are provided and have been suitably checked against specifications. So this includes ensuring that we have reference for each test method, including version numbers, statements indicating the weight or measure of the sample, data on the method of preparation and information relating to reference standards, reagents and standard solutions, ensuring the raw data for each test is made available, a record of calculations performed in connection with test results, including all the units and measures and conversion factors and equivalency factors, a statement of test data and how that compares with established acceptance criteria, a justification for any modifications to established analytical methods, and any applicable out of specification investigations need to be documented and completed. Also with batch release, it's important that all deviations that may, particularly those that impact directly to batch quality or which may signal an out of control situation, have been suitably reviewed and closed, including a statement as to the batch disposition. We also need to have the detail of the root cause and the root cause analysis and proposals for appropriate corrective and preventative actions. So the person undertaking batch review needs to assess each deviation and be satisfied that the impact on the batch under review is not impacting upon the sterility assurance. So this is where a slightly more nuanced approach is required. It's about just getting all the little bits of information, all the strands that might indicate something atypical is occurring. So developing this point as to what might be happening in the background, it's useful to recap that we need to be satisfied that we are maintaining environmental controls. And any variations with environmental controls that might impact on the batch manufacturing process need to be assessed. So inclusion of knowing that all air handling systems have been commissioned and qualified, that we haven't got drifts with airflow velocities at grade A, for example, that our air exchange volumes, as expressed in air changes per hour, are still sufficient to maintain the required room air classification and environmental conditions and can achieve the required recovery time from in relation to the respective clean room grade. And then we maintain good pressure differentials between areas of different classes. So again, drawing in that whole clean room control element is just as important as knowing about the water system as an example. So why does all this matter? Why this particular emphasis upon sterility assurance? Well, if you have a look at some recent US Food and Drug Administration warning letters, then very quickly, just looking through 5 or 6 letters, I've drawn out that sterile manufacturers have been called out on failure to reject active pharmaceutical ingredients contaminated with foreign materials, the release of multiple batches with test methods not validated for their intended uses, failure to ensure that all release lots and lots in inventory are meeting their revised specifications, having outdated certificates of analysis, failure to control, process, analyze, approve or reject raw materials, which we spoke about earlier, the release of product using non-validated processes, no supporting stability data, and perhaps in this sense the one that no sterile product manufacturer wants to receive, and that's a regulatory citation saying sterility assurance or environmental monitoring is seen as weak, because in those situations it unpicks and undoes all of the elements that go into batch release. So the batch might look like it's in specification, but our controls around that are poor and sterility cannot be directly measured or directly tested for. So in summary, and drawing the main part of the webinar to a close before we move on to questions and answers, batch release is the final part of the manufacturing process, and it's the last chance to prevent unsuitable product, and by unsuitable product we're talking specifically here about patient risk, from being released into the market. One of the most important aspects during the review of an executed batch record is that we have documented any unexpected or atypical events, including what's relating directly to the product and also what's going on around our environmental, water, raw material controls and so on. And as well as assessing that the product is efficacious, sterile products assessment does bring us additional concerns, and we can only assess sterility assurance from the point of view of probability or confidence level. So that's the main parts of the webinar. As Annette says, there is a supporting white paper which goes into these points in a lot more detail, but at this stage I can hand you back over and we can just take any questions.
Okay, thanks, Tim. Thanks for that comprehensive review of the sterile products and the factors involved in the required for their release. We have had a few questions come through, so bear with me while I try to read these. We've got one from Penny. Would you expect a microbial bioburden test to be performed for all raw materials for terminally sterilized products?
Well, I think that's a good question. I think yes, to the degree that you would test microbial limits test on raw material in general. So I think it's the same for terminally sterilized products and aseptically filled products. It is possible to do some reduced testing. So one approach sometimes is taken is that inorganic materials might be subject to skip lot testing, but all raw materials, vegetable origin, are subjected to testing. And it also stands that raw materials currently antimicrobial, as will be shown in validation studies. So where certain acids are used, for example, that there's a good chance that they will be antimicrobial. But when you get into the realm of things like sugars and so on, then these can be contamination, so they should be subject to testing.
Okay, excellent. I've got one from Chris here. What are the main sources of contamination in sterile products manufacture?
Yeah, so multiple sources, people, water, faulty air handling systems, and the transfer of materials in and out of the facility. Now, most studies will show that the primary source of contamination is people. And I think looking at various bits and pieces, it tends to come out around 70%. And that's often reflected in the microbial identifications that are done, where there's an abundance of organisms linked to the human skin. The second highest source tends to come out as water, and water is has a double conundrum, because not only does water allow certain organisms to grow and reproduce, negatives will be able to. It's also an effective vector of contamination, so water go quite far. But, and then third on the list tends to be the act and material transfer. So if we're not ensuring that items are, say, triple layered with the layers removed appropriately through different stages, and if we're not properly applying, say, sporicidal disinfectants to cover those stages. So to my mind, those three probably come out as top.
Okay, excellent. Another question from another Chris. Is sterility test required for batch release of all aseptically produced products, even if each unit is individually prepared? For example, a 100 ml bottle spiked with small amount of active, repeated many times.
Yeah, I mean, that's probably something that needs to be agreed with a regulator, but if they're all produced within one session, then my understanding is that they would be classed as one batch. So it should be representative samples taken from that session, particularly near start, close to middle and near end, but it might relate more specifically to a specific, the general answer. I think so. Without knowing your process specifically, then you know, there might be scope to talk with the appropriate regulator.
Okay, thank you. I have one from Sarah. Why do batch record errors occur?
Yeah, I cover some of this. I think that there's some interesting research into this, and there's a US GMP consultant called James Vesper, who's written a bit onto this, and it's all about one of the key reasons is human design factors. It's often that a batch record might be produced by a QA department who are some distance away from the production department, and it's not necessarily intuitive for operators to complete. So batch records should be designed with the operator in mind. But I also think that records, provided they tick all the data integrity boxes, can help to reduce errors, because it certainly can't miss things out, because you won't be able to move on to the next page and that page is completed. So if you were to accidentally type in an extreme value, often they're configured that they're flag out of range values and require a response to be made at that point. So there are ways to kind of bring those errors down as well.
Okay, thank you. And I think we've got time for one more question from Leanne. Would you test endotoxin for all raw materials for thermally sterile product, or is it feasible to justify not performing this on raw material test?
Yes, it is justifiable. It very much depends on the raw material and its origin and the probability contains endotoxin, coupled with what your manufacturing controls are and whether you have endotoxin removal steps. But one important point to bear in mind is that the European Pharmacopoeia have now begun a process that every time they update raw material monograph, they're removing the microbiological attributes. So it would, in the past, it may have specified a bioburden and endotoxin test. It's down to the manufacturer to risk assess this in relation to the product or their particular process. But in terms of the endotoxin, it really does depend. So I mean, if you're using sorbitol or sucrose in a process, for example, then these do have an association with endotoxin. Using something that's got more of an extreme of a pH, or something that's particularly more viscous, oily in nature, then generally these tend not to contain endotoxin. So it's all about understanding where the raw materials come from and what the manufacturer is doing and whether there's any chance of contamination at the source of production of the raw material.
Okay, well, that's all we've got time for today. Thanks, Tim, for answering those questions. As I mentioned, any we haven't got round to during the webinar, we will answer offline and share them with the whole group. I'd like to thank you all for attending our webinar. I hope you found it useful, and we appreciate those questions that you had. I'd like to remind you that I'm going to send out a copy of the slides along with a more detailed white paper in the next few days, so look out for that. And I'd like to remind you that this was the second in our series of 3 webinars. Tim will be returning to do the third in the series, investigating sterility test failures, on Thursday the 4th of June. You can book online on our website, and as before, I'd get in early because our places fill up quite quickly. So on behalf of RSSL Sterile Manufacturing team, I'd like to thank you all for listening today. Please do not hesitate to contact me if you have any questions or if you need any support with any projects. And thank you for your time.